So you're aware that there's not enough coronavirus testing, but aren't super clear on the specifics of what we're testing for. But what if I told you that there were really two major approaches to testing, and the type that's not yet widely adopted may become increasingly important as the pandemic progresses? Also, you may have heard about giving actively infected patients serum drawn from those who have recovered, but that treatment sounds like science fiction to you. I'm Byron Liu, a hospital-based internal medicine doctor in the Philadelphia area, and I want to talk about how COVID testing works, how approaches to testing may evolve, and what this has to do with the rationale for convalescent plasma therapy. Because I'm shuttered away while I'm off service this week, I'm joined by my husband Dan, who is not a doctor, and will help me talk things through.
Hi folks. So my sweet, let's jump in. What are you talking about?
Okay, so COVID testing currently falls into two broad categories. Testing for the presence of the virus itself, or testing to see if your immune system has been alerted to its presence.
Which is more common?
The first of these, looking for pieces of viral genetic material in patients' noses, has by far been the most common way of testing. Briefly, the test subject swab is digested into bits, generating a census, so to speak, of who is hanging around the nose. Then, a copying system specific to the novel coronavirus is added to the mix. The system lights up every time coronavirus genes are copied. So let's say 100 copies of coronavirus are present in the initial swab. The first go around, you end up with 100 flashes of light, then 200, then 400. And these flashes are tiny, but by, say, the 15th cycle of copying, there will be 1.6 million flashes, and by the 30th cycle, 53 billion flashes.
I feel like we're all sadly used to exponentially increasing curves at this point.
This process is called testing by RT-PCR, for real-time polymerase chain reaction, since it takes advantage of optically capturing a chain reaction as it happens in real time. Since the chain reaction only happens when coronavirus is originally present, it is used as a diagnostic test for the virus itself. Okay, so a major problem with nasal swab testing are false negatives from sample collection. If you don't get swabbed deep enough, your test may result as negative despite being infected, which you can imagine has public health implications. The false negative rate for these tests is as high as 30 to 50%. Also, while this is useful for testing patients with COVID-19 symptoms, widespread testing from Iceland and South Korea suggests that somewhere between 1/4 and 1/2 of patients are asymptomatic.
So everyone is a potential threat. Is there a way of telling if you've already had coronavirus?
That's where testing a patient's immune response comes in, and this is called serologic testing.
What does serology mean?
You may remember from high school biology class that serum is the liquid part of blood, as opposed to the red cells and white cells that are heavier and would sink to the bottom of a test tube if you spun it really fast. Proteins like antibodies are found in serum, which, to talk like a vampire, is the brothy part of your blood. Serology refers here to antibodies in the serum.
How do these antibodies show up?
So in response to infection, B cells, a type of white blood cell, mass-produce antibodies called immunoglobulins, like they're little weapons factories. These can neutralize disease-causing viruses or bacteria and tag them to make it easier for white blood cells to capture them, as well as a crazy cascade of other responses.
And so we're looking for these antibodies? Ones directed against coronavirus?
Yes. Testing for the presence of these coronavirus-directed antibodies is typically done using a system called ELISA. Which sounds very pretty, but is really complicated and would take me more than 10 minutes to go through. But long story short is that ELISA uses engineered light-up antibodies to check for the presence of other antibodies.
Antibody inception.
Sure. So up until now, I've been drawing antibodies as this one stereotyped Y-shaped protein. But really there are 5 classical types of antibody, or immunoglobulin. But we're going to focus on just 2 of them, IgM and IgG. IgM is traditionally thought of as the fast-acting antibody. Even though it's on the bigger side and looks complicated, it can be made by your B cells relatively quickly with minimal training.
So it's kind of like shrapnel or a ninja star. Relatively easy to make?
Yeah. But despite this, it still takes about a week for your body to kick production into high gear. The trade-off for the relative speed of IgM production is that it doesn't neutralize the enemy as effectively. Also, after the initial response to the infection is over, IgM production stops.
So how about IgG?
IgG production typically takes even longer to rev up. IgG attacks targets with intense precision, and because of its small size, can be produced in absurd amounts.
So IgG sounds more like a tiny guided missile.
Yep, it's super effective. But due to the specificity, the B cells that produce it typically require extra training steps. But production of these antibodies continues for months to years and can confer lasting immunity. A subset of your B cells that has undergone training persists and gives you immunologic memory, which means that they can activate at a moment's notice next time.
As a tangent, is that why most people usually can't get chickenpox twice?
Yeah, because people who've been infected have memory B cells that produce chickenpox-directed IgG for years. These guys can activate without training again the moment chickenpox is detected by the body again. This is actually the most basic way that vaccines work.
Do we test serologies like this for other diseases?
Yes, we do this for things like HIV and hepatitis B or C. In fact, for viruses like hepatitis B, which can have really oddball behavior, the type and target of immunoglobulins is important. For example, due to the specific timing, we often assume that detectable IgM implies recent acute infection, whereas IgG, since it persists, implies infection at some point in the past.
Are these timeframes true for coronavirus?
So I don't know. While these are the classically taught timeframes, in the original 2003 SARS classic outbreak, based on the one paper I found, IgG and IgM levels seem to have both reached high levels at the same time, which is obviously non-standard. So we're not quite sure yet how this one behaves.
So why don't we just test for IgM for coronavirus?
Remember the roughly 1 week period where you have an infection, but IgM production hasn't ramped up yet? We, in the biz, call this green area the window, which occurs before seroconversion, a term that describes when antibodies become detectable on testing.
So I guess IgM can be used, but the risk is that testing IgM too soon will result in a false negative. Got it. But from a test collection standpoint, are there the same problems with false negatives as there are with the PCR swabs?
Spoiler alert, in medicine there are always problems with false negatives and false positives. But since serologic testing is a blood draw and you're clearly able to see if there's enough blood in the test tube, it skips some of the nagging questions about sample acquisition that we have with nose swabs.
So is IgG testing going to be more important going forward?
Well, that's one of the big questions, is whether infection with SARS-2 coronavirus induces an IgG response that is protective. You can check levels of IgG all you want, but if that doesn't correlate with immunity, it's less helpful in making decisions. Take HIV, for example. Having positive IgG doesn't make you immune to HIV.
What kind of decisions does this guide?
One example is hospital staffing. Would you rather have a healthcare worker who's immune to the disease already, or someone who's at risk for new infections staffing your isolation units? Testing for healthcare workers can help guide decisions on who works where and how scarce protective gear is allotted.
Okay, but again, none of that matters unless IgG levels are proven to be linked with practical immunity.
Right, and while it makes sense that this would be true, I think it's dangerous to just assume this without good studies. Another good example besides HIV is hepatitis B. Remember that in addition to the type of antibody, IgM versus IgG, the target is important. For example, IgG directed against the hepatitis B core protein implies prior infection, but offers no immunity. In contrast, IgG directed against its surface protein does confer immunity.
So we have to hope for SARS-2 coronavirus. Whatever IgG currently being tested for definitely implies immunity.
Yep.
Okay, so what's this business about giving immunity from one person to another?
So in this case, what you're describing is called convalescent plasma therapy. Convalescent is just an old-timey word meaning recovery from disease. The idea is to take advantage of somebody's antibody-based immunity. Remember how if you've recovered from COVID-19, you already have B cells splitting out large amounts of IgG directed at coronavirus?
Yes, and this IgG gets to high levels in the bloodstream later in the infection and is extremely good at homing in on the coronavirus.
Right, so this is like an inter-patient arms deal. Donor patients undergo a process called apheresis, where blood is drawn with a big catheter. Nothing here is to scale, by the way. Big chunks from your blood can be filtered out, leaving proteins, including antibodies. Again, using ELISA-based techniques, specific SARS-2 coronavirus-directed IgGs are selected out of this soup and given to recipient patients.
Does the timing of the donation matter?
Yeah, so even though IgG persists for months to years, it's highest right after the infection. So harvesting a good amount of it is easiest in donors who are actively recovering or convalescing.
But aren't actively infected recipient patients going to be making their own IgG eventually? Why don't we just wait for that?
Remember how exponential growth works. Giving a bit of theoretically effective IgG a few days early before the recipient's own immune response has kicked in can forestall production of tens of trillions of viral particles and keep the patient below the threshold where the virus kills them. You are, on a microscopic level, trying to crush the curve.
Are there any studies backing this up?
A small group of 5 patients in China, all on ventilators, were treated this way. All 5 people had some level of recovery, but 5 is a tiny sample size. Bigger studies are underway in New York, and I suspect results will be out, tomorrow.
Well, this has been helpful to think through things. As someone who's not a scientist or doctor, it's nice to know the basic mechanics of why things are being studied.
So, all you cool cats and kittens, we had to cut out a ton of details about the nitty-gritty of these processes, but the basics are there. This video is accurate as of April 3rd, 2020. Papers referenced are posted in the video description below. And as they say on the YouTubes, if you like this, please share, like, and subscribe.
This has been a production of I Am Sketchpad.